So we've discussed cells and all the great things cells can do with their organelles, and now I want to discuss where are cells getting their energy. So before we can talk about that, we need to discuss what is energy. Energy is the ability to do work, and this can be mechanical energy like riding a bicycle, electrical like you see in lightning, or radiant energy like we can all feel from the sun. Now, when we discuss energy, there are two forms that all energy comes in that you need to understand. This is kinetic and potential energy. Potential energy is stored energy, energy that's available for use. Kinetic energy is the energy of motion, or energy that is currently being used. So consider a roller coaster scenario. At the top of the hill you have potential energy, and as you go down, it's kinetic energy. It's the energy of motion. Now, obviously, our cells are not on roller coasters going woohoo with little cytoplasm arms stuck up in the air and screaming. So, where are our cells getting their energy? This is an amoeba we have here. How does this amoeba get energy in order for its organelles to function? Cells use chemical energy, and in this case, chemical potential energy is how the energy is stored. And here's an example of a hamburger. Now, cells do not eat hamburgers, and amoebas definitely don't eat hamburgers, but they do feed. All heterotrophs, which are organisms that cannot produce their own food, have to get their food from somewhere. And amoebas do this through a process which is called phagocytosis. So let me show you a brief video of an amoeba consuming parameciums through phagocytosis. You'll see it's reaching sort of some arm-like cytoplasmic forms around the parameciums. These are called pseudopods. The amoeba has captured the parameciums, and this is how an amoeba gets food. And this is the true life and death struggle of the microcosmic world. So now you've seen the amoeba's version of fast food. So how do we get from the food we consume, whether it be a paramecium or a hamburger, how do we get that into energy our cells can use? Well, it has to be broken down into its basic subunits, such as carbohydrates, fats, proteins. Energy can then be released from the bonds in these chemical components, and this can be stored in various ways, creating chemical potential energy. And then it can be used in the cell as kinetic energy as processes require it. There are good sources, obviously, that are much better than a hamburger for chemical potential energy. My husband actually insisted I throw this in here for you. So here are some good, healthy sources of chemical potential energy: milk and yogurt and fruits and vegetables and lean meats. Now, we're going to back up and talk about some basics of chemistry for just a minute, but don't make a face. This is much scarier than chemistry. See, now this equation doesn't look so bad anymore. So we're looking at the pieces of a chemical reaction. The parts that you start with are called the reactants. In this case, we have hydrogen molecules and oxygen molecules, A and B. What you get after the reaction has occurred is called your product. In this case, we have water molecules. So these reactions can go forwards and backwards, but it's important to know those basic terms. So what holds molecules together? It is the bonds. And what do atoms exchange to share when they bond? They share or exchange, drumroll please, electrons. And remember, electrons are what all bonds are made of. It's the sharing or the giving of electrons between atoms that causes them to stay together. And the same is true of molecules. It's all about the chemical bonding and the electrons. So when molecules break and form bonds, electrons are being exchanged, and electrons are energy, so energy is being exchanged. And energy cannot be created or destroyed according to the first law of thermodynamics, so that energy must be going somewhere. So now let me torture you for just a few more minutes with some chemistry images, and this is a graphic representation of a chemical reaction. This is an endergonic reaction. Now, if you'll think back to the terms I had you define earlier, ender means inside or within. So this is a reaction that will be taking in something, and it's taking in energy. So if you look around along the x-axis, or the bottom line of this graph, you'll see the progress of the reaction. This is the process of going from the reactant to the product. Along the y-axis, the vertical line, you have potential energy of the molecule. This is the amount of energy that is stored within the molecule. If you look at this graph, when we start off with our reactant, there is very little energy stored. By the time we get to the product, there's a great deal more energy. But what is with this hill thing? Some energy is lost as light or heat during the process of this reaction. So actually more energy is put in than is successfully stored in the final product. So there is some loss of energy. Now, in an exergonic reaction, exer means to leave or exit. So energy is coming out of this reaction. In this case, the reactants actually have more energy than the product. But again, we have this funny hill. This is actually an activation energy now. You have to put energy into an exergonic reaction in order to get the energy out. You have bonds that have formed in this molecule, and in order to break those bonds and release their energy, you have to have something go in there to start shaking these bonds up and causing this molecule to change and react. This amount of energy that is required to start breaking these bonds is called the activation energy. So cells will be able to store energy within their chemical bonds, then they can release it from the chemical bonds with endergonic and exergonic reactions. Now, another type of reaction we need to discuss are oxidation and reduction reactions, also called redox reactions. During the oxidation of this portion, you will have an electron get donated. The donor of the electron is referred to as being oxidized. The molecule that accepts the electron is called reduced. And yes, I know this sounds completely backwards. I did not say this wrong. During oxidation, a molecule loses an electron. The reduced molecule has gained an electron. So your electron acceptor is reduced. Your oxidized molecule loses an electron. Your reduced gains an electron. I know it sounds crazy, but you will need to be familiar with the idea that the passing of energy can be represented in reference to a redox reaction, and that the oxidized agent has lost energy and the reduced agent has gained energy. Now let me just briefly touch on a famous redox reaction, and this is one you will hear plenty more about in our next unit. It's photosynthesis. It is a redox reaction. You start off with your reactants being 6 molecules of carbon dioxide, 6 of H2O, which is water, and some energy. And this yields a molecule of C6H12O6, which I really hope you all know is glucose, and 6 molecules of oxygen. Water, in this case, is oxidized. It loses a hydrogen ion, or H+, and an electron. And CO2, or carbon dioxide, is reduced, gaining the hydrogens and the electron. That's how we end up with C6H12O6. We're adding the hydrogens into our carbon dioxides from the water. Respiration is also a redox reaction. It is the opposite of photosynthesis, and again, you'll hear plenty about this later. Here you take the reverse reaction. Your products have now become the reactants. C6H12O6 is combined with 6 molecules of oxygen, and this produces 6 molecules of carbon dioxide, 6 of water, and some energy. Glucose in this case is oxidized, and the oxygen is reduced. The energy release goes into the electron transport chain to produce ATP, and I mean the energy that is released during respiration. Please do not panic over the electron transport chain right this minute. We will get to it by the end of this lecture. We'll talk about it more next week, but for the time being, just know that that is one place the energy from a redox reaction, or more specifically from respiration, can go. Now the final type of reaction that I want to spring on you today, or the final pairing, is the condensation reaction and the hydrolysis reaction. Now if you think about condensation in the morning out on the grass, you're picturing water on the grass, and that's exactly what a condensation reaction does. Your reactants come together and they produce a product and water. Hydrolysis reactions go in the opposite way. They go from a reactant in the presence of water, and it produces a different product, ENG, but it takes water to break apart your initial reactant to get your products. So just to clarify, in a condensation reaction, water falls out of the reaction. There's condensation. You think of water falling out of the air when dew forms. And hydrolysis, remember hydro means water, lysis means break. So in hydrolysis, water breaks. Water breaks the bond, breaking apart a molecule. So typically in a hydrolysis reaction, you'll go from a single reactant to two or more products. Alright, so by now you're probably feeling a little bit misled. I started off this lecture saying we were going to discuss how cells get their energy, and that went just huge chemistry spill. But so how do these different terms, endergonic, exergonic, oxidation, reduction, condensation, and hydrolysis, how do they all relate back to that chemical potential energy in cells? Well, the chemical potential energy storage in cells can take two forms, as long-term or short-term storage. Talking about long-term, mostly lipids, but carbohydrates as well, sugars and fats can store energy. When you get to short-term energy is where it gets really interesting and what I want to focus on today. This is where we have ATP, or adenosine triphosphate. Don't panic, we'll get back to the ATP in a second, but just know it's a way of storing energy from chemical bonds in the short term. This is where all these new terms will come into play. I need you to know these terms so you'll understand what I'm talking about when we talk about ATP. In addition to ATP, there are electron carriers called NADPH and NADH. We will talk about these more later when we get to photosynthesis and respiration. But for now, just know that they are electron carriers, which means they can store energy. But like ATP, these are short-term energy storage. From where we break apart the molecule, then we shuttle the energy over to where it's needed. It is not stored for any amount of time. These are just energy transport vehicles. Now, let me go ahead and move you on from this discussion of chemistry into how we are taking this chemical potential energy and moving around the cells. And it starts off with ADP, or adenosine diphosphate. Now, adenosine diphosphate is made of three main components. You have an adenine in your top right, you have a ribose sugar there in the middle, and you have two phosphates, which have the P's on them now. And I'm going to clean this structure up a little bit for you. Let me just, there we go. Here is your adenosine diphosphate. Di means two. Adenosine is how I refer to the ribose and the adenine grouping. And so this is how you're going to see it represented. There's your adenine, your ribose, those make up adenosine. Two phosphates is diphosphate. And then you've heard me say adenosine triphosphate, and that just has three phosphates. Like a tricycle has three wheels, triphosphate has three phosphate groups. So what we're going to do is go from ADP, or adenosine diphosphate, to ATP, or adenosine triphosphate. Now, here's where the chemical potential energy comes in. ADP is a lower energy molecule. What we can do to store energy is to make it into ATP, which is higher energy. And that energy is stored within the bond between the second and third phosphates, that phosphate that takes us from ADP to ATP. We're cramming energy into that bond, and that's why you see the energy symbol here above our yields, or our arrow symbol. So what we'll do, we take our ADP and an additional phosphate, and we need energy to make this happen. So if we're putting energy in, energy goes in, it is endergonic. So we cram this last phosphate on, we add some energy, it takes energy to do this, and then energy is stored within that bond. And yes, it does take a little more energy to do this than is actually getting stored, so you are losing some energy. Once we've pushed that energy in there and attached the phosphate, we will have adenosine triphosphate and water. Now wait a minute, was there water originally? No. So if water falls out of the reaction, what kind of reaction do we have? A condensation reaction. And there's one thing I forgot to mention before is phosphorylation occurred. What is phosphorylation? Well, phosphorylation sounds like phosphate, and that just means that a phosphate has been added. So phosphorylation has occurred going from ADP to ATP because we went from 2 phosphates to 3 phosphates. So we had phosphorylation. If you'll think back now to when I discussed reactions and your products and reactants, I mentioned that reactions can go both ways. So here I flip the arrow around to take us from ATP and water back to ADP and phosphate and energy, and this releases the energy. So when energy exits, you have an exergonic reaction. Here, there's no water coming out of the reaction. Water is one of the initial reactants, or it's involved in the reaction there over the arrow. It breaks the bond between your last 2 phosphates, and so it is a hydrolysis or water breaking reaction, and then dephosphorylation occurs because we have taken a phosphate group off of ATP when we produce ADP. So going from ATP to ADP, we actually can have phosphorylation occur because not to ADP itself, but to another molecule because this phosphate group has been released. Adding the phosphate group to another molecule may change the shape of the receptor molecule, and it may energize the molecule, causing it to break bonds and fueling an endergonic reaction, which takes in the energy that has just been released from the ATP. So this movement of phosphate groups is key to following the energy. When you add a phosphate group on, you are putting energy into a molecule. When the phosphate group comes off, energy is being released. So if you want to add that phosphate back on somewhere else, that energy can be used to reattach that phosphate and energize another molecule and another reaction, and that new one would be endergonic. But going from ATP to ADP is exergonic because energy is released. This brings up a few questions. One of which is, what stops this reaction from being a cycle and reaching chemical equilibrium? Why don't we just go from ATP and water to ADP and phosphate and energy, then have energy and ADP and phosphate turn right back into ATP and water? Why doesn't it just go in a constant cyclical cycle? Because what happens is cells use the energy quickly by utilizing it in another reaction. So that energy that comes from when we go from ATP to ADP, that energy that is released is immediately used with another reaction. This is called coupling reactions. You take an exergonic reaction, such as going from ATP to ADP, which releases energy, and it occurs with an endergonic reaction like protein synthesis that needs to take energy in order to create a new molecule. So ATP provides the energy needed to make protein. It also provides the energy for many other various cell functions, but this is an example. But a coupled reaction is what prevents chemical equilibrium from occurring, and just that constant cycle from ATP to ADP to ATP to ADP from occurring. Another question you might have is, why can't you just break down ADP into AMP or adenosine monophosphate to release energy. Why do we have to have 3 phosphates? Why not just go from 1 to 2? Because there is not as much energy in the bond between the first and second phosphate groups as there is between the second and third. So if you can think back to being a kid on a school bus, if you can picture being a child on a school bus, if you never rode it, if there's 2 of you in the seat, you're fine. You're comfortable. You can both sort of shift your book bags to one side of each other, and you're not bumping into each other constantly. Now, cram a 3rd kid in that seat and put yourself maybe in middle school. How crowded is that? And how agitated is the kid sitting in the middle? There's not enough room for them to move. They can't shift. Every time they move an elbow, they're bumping somebody. So it's a lot easier for them just to push that 3rd child right off the seat. And they can spread out more comfortably. So there is more energy. There's all these molecular components that are bumping into each other, and that there is, when there's less space for them to move, such as between the second and third phosphates, it takes more energy to force that 3rd phosphate in, because it's like a kid going, no, 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 I don't want to sit there. Then when you release it, there's more energy to be released because it took more energy to get it on there, so there's more energy tied up in that bond. Because this bond is high energy and so much energy can be stored in that bond, ATP is considered the energy currency of the cell. If the cell, say one organelle, needs a bit of energy, it calls in for ATP and makes a quick energy withdrawal from this cell. This comes out initially, of course, with the food we eat, and that food is broken down into components, and then the molecules are broken apart to get energy to produce ATP. And to do that, we have to go back to our pesky electron transport chain. So, we're going to cover this briefly now, in more detail later, but the electron transport chain is a series of membranes bound carriers that pass electrons down through a chain of redox reactions, which release energy. Remember, redox reactions, you're passing electrons from a donor to a receptor. This energy is used to power transfer proteins to move hydrogen ions across the membrane against their concentration gradient. Remember, transfer or carrier proteins change their shape, and they can move material that couldn't usually go through the membrane with or against their gradient. In this case, hydrogen ions are going against their gradient. The buildup of hydrogen ions is then released through an enzyme called ATP synthase, and this is set up much like a channel protein, like a spinning combine on the end, or not like a water wheel on the end. ATP synthase is an enzyme that uses the hydrogen protons going down their gradient and releasing energy when they do this to power ATP production. This process, when ATP synthesis uses the proton gradient to produce ATP, is called chemiosmotic phosphorylation. Now, even though I'm sure you now all have a great grasp of the electron transport chain, or the ETC, and chemiosmotic phosphorylation, I'm going to talk about it just another minute, just to clear up any questions you may have. So this white represents a membrane. These are membrane-bound carriers that I mentioned before that pass the electrons down through a redox reaction, and they are what make up the electron transport chain itself. Also, in the same membrane, you will have your ATP synthase. As I mentioned, it is sort of like a protein channel with some sort of water wheel attached to the base of it. This is your ATP synthase. It is an enzyme. So, what will happen, and just to make sure we have a key here, little yellow dots are going to represent electrons, which electrons are energy. And then we have electrons that will be transferred down the electron transport chain by redox reactions, and as they go down, little bits of energy are being released. This bit of energy is going to be used to pull in hydrogen ions. We're going to represent those by this reddish pink color. We're going to take hydrogen ions and going to pump them using those carrier proteins into the membrane. And we're forcing them in and making it more and more and more full of these little hydrogen protons. So after this energy or these electrons have gone down the electron transport chain, our cell membrane is going to be stuffed full with hydrogen ions, and it will look something sort of like this. Let me slap on another ATP synthase real quick. Alright, so like I said, it is chock full of hydrogen ions. Now, the concentration gradient is such that there is a higher concentration inside the membrane than outside the membrane. So like people in a crowded room, what are those ions just dying to do? There's lots of pressure on the membrane and they're pushing and they're struggling and it's so crowded. They find that channel through the ATP synthase and start scooting out. And because there's pressure behind them, it releases energy as they come flying out of there. And that energy is released. Now, is the cell just spitting out little pieces of energy to fly around? No, it's going to use them for something, and it's going to use them to take ADP and that inorganic phosphate. It's the energy that is combined with those to produce ATP. So this is where we get our ATP, is we create that proton gradient by bouncing energy down. We force all these hydrogen ions in, and energy is released when they escape through the ATP synthase, and the ATP synthase takes ADP and phosphate and uses that energy to cram that last phosphate on the ADP, and then you end up with ATP, adenosine triphosphate. There's your adenine, your ribose, and then 3 phosphates are just crammed on there, and it's a high energy molecule. And this is how we get ATP, or this is one of the ways we can get it. So, the energy released by the electron transport chain is then stored in chemical bonds in ATP. And this is really, really important. And this process is called chemiosmotic phosphorylation. You're adding a phosphate, it's phosphorylation, and it involves the pressure because of the proton gradient within the membrane, hence the chemiosmotic. And so we call it chemiosmotic phosphorylation. The proton gradient fuels the ATP production using ATP synthase. Alright, so the key points that I want you to get from this lecture are the first law of thermodynamics, kinetic energy, potential energy, chemical potential energy, the basic points about chemical reactions. I also want you to understand endergonic versus exergonic, oxidation and reduction, hydrolysis and condensation reactions, the relationship between ATP, ADP, and energy. I understand chemical equilibrium and coupled reactions, the electron transport chain, ATP synthase, and chemiosmotic phosphorylation. I promise all this material will start to make a lot more sense very soon. You're going to see how it all applies to cells and specific cell organelles. But for right now, focus on the idea of ATP being high energy and how we get that chemical energy. Where is it coming from, and how do we get it there ready for the cell to use? If you have any questions, as always, please feel free to contact me. Don't forget, we do have Yahoo Messenger now. Please see the course website for the contact information, and I look forward to hearing from all of you very soon.